this juncture, I also would like to welcome our Tamil Nadu Medical Council observer, Dr. T. Prasad, who has joined our session. Welcome, sir. Thank you, sir. Now I request the moderator of the next session to take over.
Okay, good. So, dear delegates, welcome back for the next session. And I would like to introduce the chairperson for the next session. It is Dr. R. Vinod, who is the professor and head Department of Microbiology, head of the Virology Lab, and vice principal of Sri Venkateswara Medical College Hospital and Research Center, Pondicherry. He is having 15 years of teaching experience and expertise in the field of microbiological diagnosis. And under his able leadership, Sri Venkateswara Medical College Hospital and Research Center obtained NABL accreditation for the BSL-2 Virology Lab. He's having around 20 publications and guided around 15 MD students, and he has many ICMR STS projects to his side. He is a question paper setter for many universities like Dr. MGR Medical University, Rajiv Gandhi University of Health Sciences, Vinayaga Missions Research Foundation, etc. He is a reviewer for many journals like JCDR, IJCBR, etc. Dr. R. Vinod, welcome to our session, and I really thank you for accepting this to be the chairperson for this session. Over to you, sir.
Thank you, Dr. Manju. Am I audible?
Yes, you're audible.
Good morning to one and all. I thank Vinayaga Mission University and BMSCON for giving me this opportunity. I thank them. And I would like to appreciate the speaker and the organizing team for thinking of COVID-19, SARS-CoV2. Because it is slowly becoming a history. COVID-19 is becoming a history. There is no fear. Nobody talks about COVID nowadays. Nobody wears mask. Nobody wants to talk about a vaccine. So I hope this COVID-19 will remain a history like our demonetization. So with pride and immense pleasure, I would like to introduce our esteemed speaker, Dr. Kannan Vaidyanathan. Sir is presently the professor and head Department of Biochemistry, Believers Church Medical College Hospital, Thiruvalla, Kerala. Sir has done his MD in Biochemistry. He has also got his postdoctoral fellowship in microbiology and cell biology from Indian Institute of
Dr. Vinod, your voice, you're muted. Dr. Vinod?
Kochi.
Yeah.
From 2019 to 2022, he has headed the Metabolic Disorders Laboratory. He was a Deputy Technical Manager of Amrita Laboratory Services from 2006 to 2013. He has been the Professor and Head Department of Biochemistry and also was a Deputy Medical Superintendent in Pushpagiri Institute of Medical Science, Thiruvalla. His professional achievements and awards. He has gained Asia Pacific Federation and the Clinical Biochemist Silver Jubilee Scholarship in Beijing, Chennai, in China in 2007. He has also been awarded KP Sinha T.S. Krishnan Award for the best original research article in Indian Journal of Clinical Biochemistry. Along with the other responsibilities, he's got some more responsibilities. He is the managing director of Indian Journal of Clinical Biochemistry. He is an executive committee member, Association of Clinical Biochemists of India. He is a member of Task Force for Newborn Screening, member PG Board of Studies (POAS), member of American Association of Clinical Chemistry, American Society of Human Genetics, European Association of Cancer Research. He's got a vast research experience. He has been a co-author for 3 textbooks in biochemistry, more than 85 publications, including 25 in PubMed Index journals, deliver more than 60 lectures in 5 continents. He has guided 6 PhD students and more than 50 postgraduate students. He has been examiner for MBBS, PDS, MD, PhD, and allied sciences for various universities in India. Currently, his area of research is inborn errors of metabolism, clinical chemistry, and molecular biology. We welcome you, sir, for your talk. And sir will be talking on biochemistry of SARS-CoV. Welcome you, sir.
Sir, for the kind introduction. Am I audible, first of all?
Yes, sir, you're audible, sir.
And thank you, Dr. Manju and the organizing team from Vinayaka Mission College to ask me to deliver this talk today. I'm really grateful for my being able to be able to present today.
Our pleasure, sir.
Because of 2 reasons. One, I couldn't attend the last session because I was not in station. And the second one, because last couple of days I was struggling with a bad health condition. And I was just came to the office today just to give this presentation. So
Thank you, sir.
I hope I'll be able to convey what I want to tell. And so the other thing also, like I was hoping to have more students to listen to that. I don't know how many students are there, but it's okay. I'll so because lots of aspects are there regarding, like Dr. Vinod was telling in the introduction, what is the relevance of COVID today? Now, one of the things which I want to mention is that even though COVID is largely like gone from the universe as of now, as most statistics show, but still it's important that with pandemics like this, or it is the information that we gain from that should be used for the future prevention of further epidemics to come as well. So that is in fact one of the relevance of having this topic. So with that brief introduction, I'll go to the presentation. Is that visible now?
Yes, sir. It's visible, sir.
Okay, thank you very much. And I'll start with a brief introduction to the coronaviruses as I before going to the biochemistry of COVID. Now, there are a lot of things to be talked about regarding the coronaviruses as well as COVID and the biochemistry of COVID. So it is not possible to cover everything in the limited slot of 30 minutes because the topic is so vast and lots of research has been done. So I'll try to limit it to the basic things and leave the rest to any discussion or further interactions later on or some other time. So essentially coronaviruses are, we all know it's a group of related RNA viruses, and it can cause diseases in mammals as well as birds. And they cause respiratory tract infections, which may be mild or even lethal, as in the case of COVID-19 in some cases. And mild illnesses can include some cases of common cold, and lethal varieties can also be there, like which cause the MERS as well as SARS and COVID. And in some animals, it causes some mild infection, mild diseases, like for example, cows and pigs that have caused diarrhea, while some other diseases have caused more severe illnesses in some other species. And that's the family and subfamily of coronaviruses, the subfamily Orthocoronavirida in the family of coronavirus, and the order and the etc. Their envelope viruses essentially and positive and single-stranded RNA genome it has, and it has a nucleic acid with a helical symmetry. Now, I'll go more into the details of the genome later on, but essentially it is one of the largest among the RNA virus family. And the name is because of the characteristic spikes which are seen projecting from the surface, and which is likened to the solar corona, and that is why the name coronaviruses came. Now, if you look at the history of the coronaviruses, the earliest reports of coronaviruses came in animals in this century itself and from North America, which have high mortality rates of 40 to 90%. And the virus was known as the infectious bronchitis virus at that time. And later in the 1940s, two more animal viruses were discovered, which were related to the now known coronaviruses. And the human coronaviruses were actually discovered only in the 1960s. And of course, the other human coronaviruses discovered include the SARS-CoV virus, and in addition to the SARS-CoV2 virus, which was discovered in 2019, and the other coronavirus which caused the other infections, which I'll be coming to later on. But essentially, most of these infections have been seen. A lot of these infections have been associated with the animal coronaviruses, which have been discovered since the 1960s. And we know even in the case of the human coronaviruses, many of these infections originated in animals. Now, the structure, if you look at the general structure of the coronaviruses, they have a large, roughly spherical particle with unique surface projections. The size is highly variable with an average diameter of 80 to 120 nanometers, and even extreme sizes are also known. They have a high mass of 40,000 kilodaltons and enclosed in an envelope embedded with a number of other molecules as well. And they have the characteristic lipid bilayer envelope, the membrane protein, the nucleic acid, etc., which are characteristic of the coronaviruses. And the viral envelope especially has a lipid bilayer in which the membrane proteins, the envelope proteins, and the spike structural proteins are anchored. So that is inside. And the E and M proteins are the structural proteins, and this combined with the lipid bilayer gives a shape to the viral envelope, and it maintains a size as well. And the S proteins are essentially needed for interaction with the host proteins, and that is also important in its pathogeny. And the human coronavirus NL63 is one which is peculiar because of its M protein, which is and not the S protein. So there are some variations in the difference in between the different types of coronaviruses. And essentially the M protein is a main structural protein and provides the overall shape for the virus. And the M protein is crucial during the assembly, budding, etc., as well as a pathogenic entry of the viral life cycle. And the E proteins are minor structural proteins which have high variability among the different species of the coronaviruses. Now, as I said in the beginning, the spike proteins are the most characteristic feature of the coronaviruses. And on an average, each coronavirus particle has about 74 surface spikes. And each spike, coming to a structural spike, is about 20 nanometers long and is composed of a trimer. The S protein in turn is composed of an S1 and S2 subunits. And essentially, one of the important subunits is the what is known as a tempress protein, the transmembrane protease serine 2, which is essentially important for entry into the host cell. The S1 proteins are the most critical components in terms of infectivity, and most variable components are also there which are responsible for the host cell specificity, including the two major domains termed the N-terminal domain, the S1 NTB, and the C-terminal domain, S1 CTB, which all serve as the receptor binding domains. And the NTDs essentially help to recognize and bind the sugars on the surface of the host cell membrane. And the S1 CTD is the one which binds to the ACE2 receptor, and it is so important for the coronavirus 2, the COVID-2 virus. And there are also shorter spike surface proteins called a hemagglutinin esterase, which are important for certain species of coronaviruses, and they help in the attachment and detachment from the host cell. And inside the envelope also there is the nucleocapsid, which is forming the multiple copies of the nucleocapsid protein, which are important for the various pathogenic features of the coronavirus. Now coming to the genome, the coronavirus contains, as I said briefly in the beginning, a positive sense single-stranded RNA genome. The genome size for the coronaviruses ranges from about 26.4 to 31.7 kilobases, and it is the largest among the RNA viruses. The genome size is also the largest among the RNA viruses. It has a 5' methylated cap as well as a 3' polyadenylated tail, and open reading frames 1A and 1B, which occupy the first two thirds of the genome. And this encompasses a replicase polyprotein, or the PP1AB. And the replicase polyprotein can self-cleave to form 16 non-structural proteins, which are known as the NSP1 to 16, NSP1 to NSP16. And the later reading frames encode the major four structural proteins and of the envelope and the membrane and the nuclear capsid. And interspersed between these reading frames, as you can see in the figure, the reading frames necessary for the accessory proteins as well. That in general is the structure for the coronaviruses. And of course, the number of accessory proteins and their function is unique and depends upon the specific coronavirus, and that is the general structure of the coronavirus. You can see the spike protein, the envelope protein, which is there on the which encompasses the inside the nucleocapsid protein, the N protein. So now I won't go into much details of the replication cycle because of lack of time, but essentially, as you can see in the inset figure, the cell entry is the most important feature which starts the replication cycle, and the infection begins when the viral spike protein attaches to the complementary cell host cell receptor. And once it is attached, the protease of the host cell cleaves and activates the receptor attached spike protein. And depending upon the host cell protease available, and the cleavage and the further activation occurs by endocytosis or by directly combining with the viral envelope in the host membrane. And after that, you have the genome's translation. And once on entry into the host cell, the virus particle becomes uncoated, and then the genome enters the cell cytoplasm. The coronavirus RNA genome is directly translated by the host ribosome, and the host ribosomes can translate the initial overlapping open reading frames ORF1A and 1B, as I mentioned earlier, of the viral genome into two overlapping polyproteins, PP1A and PP1AB. And these polyproteins have their own protease, which cleaves the polyproteins at specific sites later on. And then the next important thing is the replicase transcriptase system of the protein. A number of the non-structural proteins coalesce to form the multi-protein replicase transcriptase complex known as the RTC. And the main replicase transcriptase protein is the RNA-dependent RNA polymerase, or what's the very important RDRP molecule. And it is directly involved in the replication and transcription of the RNA. And other RNA, the other non-structural proteins in the complex assist in the replication and the transcription processes as well. Now, one of the important functions of this complex is to replicate. Of course, the one replication is important, and the RDRP directly mediates the synthesis of negative strand genome from the positive sense genomic RNA. That is one of the important features of the protein, and this is followed by the replication of the positive sense genome from the negative sense genomic RNA. So it's essentially the replication which is the important function of the RDRP. And after that, the next important function is transcription. The transcription of the viral genome also occurs with the help of the RDRP. As you can see, it is the RDRP directly mediates the synthesis of the negative sense RNA molecules on the positive sense genomic RNA. And the transcription This is followed by the negative strand subgenomic RNA molecules to the corresponding positive sense molecules. And finally, there is the assembly and release, which occurs, and the replicated positive sense genome becomes the genome of the progeny virus, and by exocytosis, it is released into the host cell, and then once released, the virus can infect other host cells. So that essentially is the entire thing which is happening once the once the virus enters into the host cell. Now, coming to the classification of coronavirus, so the coronavirus essentially can be of 4 types: the alpha, beta, gamma, and delta. And alpha and beta are the ones which affect the mammals, and the other ones which primarily affect the other organisms. And that is the phylogenetic classification of the coronavirus. And the beta coronavirus is the one which encompasses the SARS coronavirus 2. And the human coronaviruses, there are 3 main coronaviruses which have been discovered so far, which have come out so far. One is the SARS coronavirus 1, which was discovered in 2003. Then you had the MERS coronavirus, which was discovered in 2012, the Middle East respiratory syndrome associated virus. And the third one is a COVID virus, which was discovered in 2019. And coming to the biochemistry of the COVID virus, it is essentially similar to what I had described so far. Now, COVID-19 is the third coronavirus outbreak in the last 20 years, so that it makes sense to think that there can be further things which can come later on. So, including the SARS outbreak and the MERS outbreak, after that is a third outbreak which has come out. And the SARS-CoV virus 2 virus is a single-stranded RNA virus with about 30,000 bases, and it shares about 70 to 100% genetic similarity with the bad virus from which it has arisen, probably by recombination, as is the current concern. And one of the important proteins or molecules which has been important in its pathogenesis is, we all know, it's the angiotensin converting enzyme 2, or the ACE2 protein. And the ACE2 protein is expressed in high amounts in most of the human tissues, especially in the alveolar epithelial cells. And that actually is the how it is the diagram which describes how the angiotensin converting enzyme 2 is involved in the pathogenesis of the SARS-CoV-2 virus. And that's just another diagram which shows how the symptoms occur in the case of the COVID-2 virus. The ACE2 mediated binding with the TMPRSS2 protein, it mediates the release of the interleukin-6 and associated various other molecules related to the immune system, and it mediates the genesis of the cytokine storm and leading to various COVID-2 symptoms, including fever, cough, fatigue, dyspnea, shortness of breath, the sputum production, hemoptysis, etc. So most of the symptoms of the COVID-2 virus are related to the, essentially related to the cytokine storm which sets in. And that is another diagram which shows the pathogenesis of COVID-2 in more details, but again, due to the lack of time, I'm not going to more details, but essentially it is similar to the entry of the coronavirus, which I described earlier, and once the SARS-CoV-2 enters into the human cell, it mediates a lot of reactions related to the release of the cytokines and the cytokine storm, and this leads to the various symptoms related to the COVID virus infection. Okay, so what about the drugs against the COVID virus? We know that by now we know that hydrochloroquine does not prevent death or illness due to COVID virus, but it just helps to halt some of the severe symptoms related to COVID-2 virus. Neither does vitamins and minerals cure COVID-19, and antibiotics also cannot cure. Dexamethasone is actually reserved only for those with severe symptoms and should never be used for mild cases. and drinking alcohol does not protect and can be dangerous. And none of the other measures, like some of the jokes which came in between, including from the US president, like spraying and introducing bleaches into the body through other means, or do not have any protective effect. And the COVID virus can also spread in hot and humid weather, and exposure to sunlight or cold weather and snow also cannot kill the virus. So these are all well-known facts by now. So what are the proteins which are then, what are the drugs which are discovered? What are the drug targets which have been identified? There have been a few targets which were described, and this have been reviewed in some of the papers. And most of the drugs which have been against some of these, one or more of these 6 targets. One is a spike protein, that's a target 1. The target 2 is the H2 protein. The target 3 is the TMPRSS2 protein. The target 4 is a protease. The target 5 is the endosome, and target 6 is the RDRP protein. So these are the main proteins which have been targeted for the various drug discoveries related to COVID virus. So of course, lots of drugs have been experimented with, even though none of these have found a complete cure. Of course, drugs are there like remdesivir and all, which are essentially targeted against the RDRP protein. And what about the diagnostic test? Now, samples, as we all know, are drawn with a nasal or throat swab. RT-PCR is the most common test which has been used to detect the virus, even though other techniques are also there. For example, the NAT technique has been employed, as well as a LAMP technique which has been employed in some laboratories. Serology testing techniques are also there, like lateral flow assays and ELISAs, which have been employed with different degrees of success. Now, what is the relevance of the rapid antigen tests? So even though they are cheaper and faster, they're less reliable and mostly have gone out of the market now. But rapid tests are done when the virus is in rapid circulation in the community or during waves of infection. And the antibody test or the serological test can also help to detect infection in the patient, even if the patient was asymptomatic, but it cannot detect the early stages of the infection. So if the patient has had the immunity to the infection, can be found out using the various antibody tests. So these are the various methods by which antibody tests can be performed, including the rapid diagnostic tests like rapid lateral flow test and point of care testing, ELISA, which can be qualitative or quantitative neutralization assays, and chemiluminescence. All these things have been done for detecting the various antibodies. And what about the, it will be incomplete if I don't mention the other tests, or even though some of them may look like out of the sky tests, they're like sniff test, which is a 25% mixture of ethanol and water which has been used. Imaging tests, of course, laboratory, when we talk about laboratory tests, we have also to look at the CT scan to give the lung findings. And serology tests are the saliva testing, biosensors, breath tests have been introduced at some stage, including the corona breathalyzer, which can detect the aldehydes. But many of these have not found much utility later on to detect the coronavirus. And essentially, most of the tests have employed the RT-PCR or NAT or the LAMP testing has been done. And functional assays have also been introduced at some scale. So lots of tests have been introduced. And also, which has something which has come in between was the CRISPR-based diagnostic tests also, which have come in between. But many of these tests have not found have not been investigated probably further also, because at that time, by that time, our vaccines have become popular, and therefore there was not much need for further development of test problem. So, but however, lots of diagnostic modalities have been experimented with for the testing of COVID. And coming to the biochemical monitoring of the test. we have also done some tests. In fact, I didn't tell at the beginning, we have done some in my previous institution, Amrita Institute of Medical Science, also we were doing the COVID testing. And in the present institution that believers that we are doing, we have done some from the private sector. Pioneering work has been done on the COVID testing, and that is why I would consider myself to why I can present this topic. So coming to the biochemical monitoring, also we have done some work on that, even though it is not published yet. The inflammatory markers, like the cytokines IL-6 and TNF-alpha can be done for identifying the COVID-19 infection, and surrogate biochemical markers in inflammation are there, like ferritin, procalcitonin, CRP, and ESR. These have all been found to be elevated in COVID-19, but however, none of them are actually specific for COVID-19, and that is a problem essentially with the biochemical monitoring. Similarly, hematological findings include the lymphopenia, elevated neutrophil lymphocyte ratio, NLR, and platelet to lymphocyte ratio, PLR. But these have definitely prognostic potential, and that is the importance of many of these biochemical and hematological markers. And D-dimer elevation, similarly, is associated with worsening of disease and increased chances of thromboembolism, deep vein thrombosis, pulmonary embolism, and disseminated intravascular coagulation. Therefore, it is related, it is important in the prognostication of COVID-19 and not the diagnosis per se. There are other biochemical markers which inflammation can lead to multi-organ failure, and acute myocardial infarction is of course an important complication of COVID-19. And for example, you have cardiac biomarkers like troponin, the NT-proBNP, CKMB, prothrombin time, and myoglobin, which have been associated with cardiac biomarkers. And liver dysfunction is of course generally common in COVID-19 infection, and liver markers including AST, ALT, GGT, LDH, and total bilirubin have been estimated in COVID-19. Acute kidney injury is one of the major complications, and renal markers like serum creatinine and blood urea nitrogen are regularly used for the detection. And of course, you need to monitor the critical care biomarkers like blood gases and electrolytes, especially for those on intensive care and as well as those on ventilators. So these are some of the markers which are used, biochemical and hematological markers. And in fact, in the case of children, the children are equally or even more vulnerable because of the weakened immune system. And pediatric monitoring tools include the CRP, procalcitonin, and the lymphocyte count. And it has also been suggested that routine urine biomarkers like the blood specific gravity, pH, proteins, ketones, RBC, and WBC count can be useful for the identification and monitoring of COVID-19 infection, but however, these are not become very popular, even though they are non-invasive because of other tests which are now easily available. At some stage, there were suggestions of introducing these urine biochemical markers as well. So what about the testing protocol? There are lots of testing protocols which were developed during the course of the COVID-19, and these include the drive-through testing, home collection, pool testing, multi-tier testing, snapshot mass testing, as well as surveillance and population screening. All have been introduced at some stage during the control of the COVID-19 infection. Now, what is important, however, is to remember about these two basic things of any laboratory testing, that is sensitivity and specificity. Any test should ideally have the highest sensitivity and specificity before it can be introduced. Same is the case with COVID-19 also. So when we look at both of these, it becomes like a difficult paradigm how to introduce many of these tests into the routine thing. And of course, as with any other test, there are also the pre-analytical errors which had to be considered, and general pre-analytical errors as well as specific problems related to sample contamination and testing of patients, for example, who are receiving the viral therapy, etc., have also to be considered when we are looking for testing for COVID-19. And in addition to these, analytical errors problems can also contribute to error. These include testing outside the diagnostic window period, as well as various other problems related to the testing itself, including non-validated assays or lack of harmonization, instrument-related testing issues, non-specific PCR annealing, etc., can also be complications related to testing errors in the COVID-19. And essentially, to reduce errors, that is why it has been suggested that we always say that laboratory results should be checked along with clinical data, and of course, where necessary, repeatability or other use of quality control validation, etc., had to be done. And of course, training, etc., is also very important in the case of COVID. So I come to the last slide. What is the role of the private sector in combating COVID-19 and future pandemic? This is important in the present context because I personally come from a private sector, and also because there are lots of private sector institutions which are working today. Now, they can definitely assist government in diagnostic testing, especially the ICMR approved laboratories. Medical and paramedical support to the patients has been pioneerly done in the current institution where I'm working, that is Believers Church Medical College. The paramedical trained support has been provided, which can assist the government in many ways. IT related, we can do a lot of things, including creating and maintaining the databases, and manpower as well as resources sharing with government institutions. And of course, as in the various other developed countries, we can have the private public partnership in policy making, which is as of now not much known in India, but this is something which can be done, the private public partnership in policy making. But definitely we can provide guidelines. The results which we obtain and the experience which we gain can be helpful for providing guidelines for future pandemics. And of course, as is being done today, the public education through CMEs and general articles can go a long way in helping the public to understand about the pandemic and the ways it can be prevented. Not to mention the role of international collaboration and helping to get funding and future research. So that in short is what I want to talk about the biochemistry of COVID. I know there are a lot of things which I left out. Now, if you want to read more about the COVID, these are some of the references which I use for collecting some of the authorized data, which there are lots of information which is available on the internet also, but we should make sure that always that it should be credible information what we are looking at. And these are some of the sites where you can get some credible information about COVID-19. So thank you very much, and happy to take any questions. Thank you.
Dr. Vinod?
Thank you, sir, for that elaborate, clear and crisp presentation on COVID-19. So do you have any experience in COVID-19 and diabetes mellitus in your during your tenure during the pandemic? Did you see any?
Yeah, there was of course association, that is well-known fact. But since the research work which I did, which I mentioned about, I did in the previous institution, and they're hoping to publish it. So I don't want to jump into that, because it's a faculty presentation. So I was associated with that. Actually, I was not the main author for that work. I was a co-author for that work. So hopefully you will see that there. But definitely there is an association as has been found in other institutions as well. That is a strong association. In fact, hyperglycemia is one of the most important features.
Since your expertise is metabolic disorders, I thought you would have done something.
Yeah, definitely. It is unofficially, I could tell that there is a strong association. Yeah.
So, sir, I think there are no queries in the chat box for the time being, sir. So, thank you so much, sir, for accepting the invitation, and thank you so much for, in spite of your bad health, I didn't know, sir, for the past 2 days, you took all the strains and this thing to present for us, sir. And it was a very elaborate session on the letting us all go through the detailed structure of the coronavirus, the genomic variations that you see, the many myths that people have, you have clarified, sir, the target proteins of COVID viruses, and the various diagnostic tests, both microbiological, serological, biochemical, hematological findings, and their correlations, and the testing protocols. And the most important thing was the last slide, what you put, sir, that was the what are the measures to be taken by people, they can take it off, sir, for combating any future such similar, we hope such things shouldn't happen in the future, but still that slide was an excellent slide, sir. The measures that people can adopt, both the government, health agencies, everybody, to combat this, combat any other pandemics shouldn't occur, sir, but if occurs. Thank you very much, sir. Now we would like to.
I have shared with the PDF of the presentation with you, madam, in case anybody wants.
Sure, sir.
Also want, you can share.
Usually after the conferences, they messages, sir. So at that time, I'll definitely share it with the delegates, sir. It's an honor, sir. And thank you for sharing us the PDF of the presentations. Now we would like to first honor, though it's on a virtual mode, sir, we would like to honor the guest speaker, Dr. Kannan Vaidyanathan, sir, first, with a certificate and a memento, sir. Next, I would like to honor our chairperson, Dr. R. Vinod, with a certificate and a memento. Thank you, Dr. Vinod, for accepting the chairpersonship.
Thank you.
Thank you, both of you. And I think we'll wind up this session, and I'll hand over to the moderator of the next session. Thank you, sir. Once more, Kannan, sir, thank you. Thank you, Dr. Vinod.